A stranger was murdered in Miss Newell's classroom, and Jason doesn't know the motive for the crime or who the killer is. To find this out, we will analyze the DNA from each piece of evidence, but first I will show you how the GSU laboratory extracts this DNA. The crucial pieces of evidence are the cheek cells of the two suspects whose fingerprints were found at the scene of the crime, Miss Newell and Mr. Matt. Also, the hair that was found beside the body is a crucial piece of evidence. The red substance by the broken beaker, the red substance of the murder weapons, and of course the red substance beside the victim. Now let's go to the lab.
Okay, today I'm going to show you how to isolate DNA from one of our samples from the crime scene. So today we're going to isolate DNA from one of the suspect's cheek cells. So today at the crime scene, you may remember that a few students asked Miss Newell, one of the suspects, to take a swab of the inside of her cheek to get the cheek cells. So I brought that Q-tip back to the lab and I placed it into a microcentrifuge tube. And there wasn't much in there, but their cells are so numerous that I know that it's in there. And I added some 70% alcohol to bring it up to this volume. Now, what I'm going to do is isolate the DNA from the cheek cells. First thing I need to do, and this is the most important step, is to add a chemical called trireagent. Trireagent is a pinkish red chemical. It's very deadly, so you have to make sure that you don't get it in your eyes or on your skin. So I'm just going to add a small amount to the cheek cells. Okay. Then I'm going to spin it down with the centrifuge to break open the cells. The reason why I want to break open the cells is because the DNA and the RNA is contained in the nucleus, and then the proteins is contained within the plasma membrane of the cell. So by spinning it, I break open each of those membranes. So I bring it out and I have a pinkish mixture. Remember, this is trireagent and cheek cells and 70% alcohol. Now, next thing I want to do is I'm going to add some higher concentrated alcohol to precipitate the DNA, RNA, and protein from the solution. Precipitating it means that I'm bringing all of those molecules out of the solution. And it makes it cloudy. So now, I'm going to spin that again. And here comes the most miraculous part. Once I bring the tube out, I have 3 separate layers. On the bottom is protein by itself. The middle layer is RNA. And the very top layer, which you can barely see, is the DNA. And the DNA is all I want. So notice how much RNA and protein there are in relation to the DNA. So now what I do, get a new pipette tip, and I want to extract the DNA off of this top layer. So very carefully, I take the DNA off the layer. Careful not to get any of the RNA mixed in with the DNA, and I add it to its own separate tube. Then, again, I add some concentrated alcohol to precipitate the DNA out of the solution. Now, I hope that I have DNA isolated in here. I can either store this at -20 degrees Celsius for a long period of time, or if I want to see immediately if I have DNA, I can run this using gel electrophoresis. I can put this amount onto a gel and run it. And I will do that will take me about an hour, and I'll come back with the results to see if we have DNA.
I added the extracted DNA to a gel and powered it with 100 volts of electricity, and I know that I isolated DNA because I have single bands, like so. Now we can proceed with the restriction enzyme analysis of the evidence from the crime scene.